was an intro video. I'm going to try to draw today. I don't know. I was like, how can I draw this out with my lack of artistic abilities? I'm going to attempt. But it takes what we learned in the nervous system and, how can we take what we already know, creating action potentials, moving of electricity, moving of ions to now control muscles and make muscles, skeletal muscles, contract. So the intro video got up until about this point and ends right around here, going through kind of the basic parts of a muscle, just basic parts of the muscle. Now, the big picture is we want to get through, how does a muscle actually contract? So I want to make sure this works. There we go. And there are 4 basic steps, and I say basic, they're not that basic. There are 4 big steps that have to occur in the muscle for it to actually contract. So if I decided I wanted to pick up a glass, how is it that I can see something, I can tell my muscles to contract, and I can pick up something? And I'm like, all of this has to happen in there. There are things that happen at the neuromuscular junction. Now, based on its name, it's where part of a neuron meets the muscles, and it's a junction. We're going to have to excite the muscle fiber. We do something known as excitation. We excited it. Now we're going to start the contraction process. And then we actually have how your muscles are going to contract, how they're going to shorten, forming what's known as cross bridges. So it's like 4 big steps. But we're going to break it down and kind of talk about each of the steps. And I was going to say I can try to draw stuff out. Your book breaks it also down into these kind of 4 basic steps, and then they put little sub-steps in there as well. I'm going to, we're going to break it down all the way down. First, looking at what are all the things that happen at the neuromuscular junction. This is why I'm like, well, I can try to draw today. I left most of my markers the other day, or my markers in the other building. So at the end of an axon, there's my neuron. It's got dendrites. Question?
Yeah. Is chapter 10 included in this one?
I think chapter 10, I think chapter 10 is just naming all the muscles.
I was just wondering.
No, I'd have to look at exactly chapter. I think chapter 9 is all of this. Chapter 10 is actually naming muscles, which lab. That'd be all lab. The major part of a neuron, it's got dendrites to receive information. It's got an axon. Yes, there's myelin along here. But at the very end of the axon, you have axon terminals. And I'm going to focus on this big guy, this one. At the end of an axon terminal, it has a little bulb at the end. So we have an axon. It's a somatic motor neuron, which means it's going to control the somatic part of your nervous system, aka it's going to control your skeletal muscle. It's going to travel from the central nervous system via your nerves, and it's going to find a skeletal muscle itself, the end. Each axon forms several branches. There's several branches. And each axon, the ending of them, has a neuromuscular junction with one single muscle fiber. So this one is going to control one single muscle fiber. That one's going to control another single muscle fiber, and so on. But I'm going to focus on that one. And I'm going to make it probably bigger and change its shape a little bit. I'll get colorful. I feel like all your muscles in your books are always some type of pinkish, reddish, bluish. There's my muscle fiber. And at the end of your axon, there's that axon terminal. It's a little bulb-ish shape. And this axon terminal is situated midway along your muscle fiber. In between, because there is a space, they are not touching directly, there's a little gap right here called the synaptic cleft. It has a fluid in there, a gel-like fluid. But it's in this, I'm going to make it bigger. So it kind of looks like that. In the end of that axon terminal, there's a chemical called acetylcholine. And you'll see it in most of my slides, just ACH. It's acetylcholine. There's a chemical. It's just hanging out. It's really not doing anything, but it's hanging out in these little vesicles inside that axon terminal. I'll make some green acetylcholine in here. It's just hanging out. Now, we also have along the muscle membrane. Now, that's called the sarcolemma. Lemma means membrane. Anything sarco in it is a prefix that means muscle. So sarcolemma is the muscle cell membrane. It's plasma membrane. We just call it a name called the sarcolemma. But along these folds, this is all part of the muscle membrane, all of it, all of that white line there are various types of acetylcholine receptors. That, yes, their job is to receive and recognize acetylcholine. Those are kind of the main players at your neuromuscular junction, where the end of the neuron is going to control that one muscle. Now, what's going to happen is that if I decided I want to pick something up, I wanted to pick up the glass, I have to tell my skeletal muscle, I can control my skeletal muscle. But I have to send some type of electrical signal, a nerve impulse, along my axon, which is going to go to that axon terminal. This little pink line, and I'll have things come up in a minute, that's your nerve impulse that's traveling along that whole axon all the way to the end of the axon terminal. What's going to happen is when that electrical impulse gets to the end of that axon terminal, it's going to open up some calcium channels. I want a different color. Oh, I have pink. That's not much different than red. Yep, that's all I got for colors. That electrical impulse is going to open up these channels. I'm going to have positively charged calcium. They're going to go into that axon terminal. Once calcium goes in, it's almost like the axon terminal is like, oh, there's just too much stuff in here. I should try to get rid of something. And it's going to release acetylcholine. So when calcium goes in, acetylcholine is going to get released. It's all these little green circles. It's going to get released and go across the synaptic cleft. It's going to find with one of these little receptors on there. Again, there's little tiny green circles. That's all your acetylcholine. It's going to create some type of action potential or electrical signal right there. So we're moving some ions in. We're going to release acetylcholine. It's going to bind with an acetylcholine receptor on the muscle membrane. Basics. Here's my big picture of it. So that action potential, that pink line that's coming in, that's the electrical impulse of me telling me I want to control my muscle. That electrical impulse is going to go to the end of the axon. Like, we're going to have some calcium channels open. They're going to go in. Calcium's going to go in. The calcium going in is going to cause acetylcholine to get released out of that axon terminal. It's going to go across that synaptic cleft and find an acetylcholine receptor on the muscle membrane. Once it goes across and it binds with that receptor is going to open up. Now we're going to have moving of more things. We're going to have moving of sodium in to the muscle cell. And we're going to have potassium moving out. Now, that means we have sodium moving in, and we have more sodium that moves in than potassium moves out. They're both positively charged. If we have something positively charged going in, and more of it in than out, what's going to happen to the charge inside the muscle cell? Something positively charged goes in, it becomes more positive. And I'm like, or less negative. Did you have a question? It opens. Yep. So normally it's not open. It stays closed. I don't know if I've got a picture. No, that they're attached there. And I'm like, so it's going to open. And you're going to have lots of sodium going in. Now, we just got done talking about action potentials in this last unit. That's what starts off an action potential. Some of those sodiums sneak in. And I'm like, and it causes a change. It becomes less negative, more positive. We're on the way to becoming a full-blown action potential. Now, we don't want these to stay open forever. We don't want these little acetylcholine receptors to stay open forever. We want them to stay open for a short period of time for just enough sodium to go in to change that positive charge, make it a little more positive for a short period of time. Then we have another enzyme that hangs out in our cells called acetylcholinesterase. Most enzymes endinase. And I'm like, and its main job is to break apart that acetylcholine. Once the acetylcholine's broken down, and I'm like, that receptor closes right back up. But it did its job. We got more sodium in, and we created that slightly positive charge inside the cell. It's the basics of what happens at the neuromuscular junction. We haven't contracted a muscle at all yet, but what we've done is we sent an electrical impulse to the muscle. I'm like, we've got a slight electrical charge that just happened because that acetylcholine opened, causes some sodium to go in, and we have a slightly positive charge. But we're getting there. Now, my summary events, because this is the big first step. The summary events, the nerve impulse goes to the end of the action potential, or the axon terminal. We released acetylcholine across the synaptic cleft. It found an acetylcholine receptor. So it diffused, bind with the receptor, and I'm like, caused sodium to go into the muscle cell, and we created kind of a small electrical event. We're on our way to be generating a full action potential. Now, I already mentioned it, but yes, we don't want acetylcholine to be hanging out forever, keeping those things open, so we've got the enzyme that breaks it down. I'm not going to watch the whole video clip. There we go. Now, we've moved some sodium in. That's kind of the end of the step of the neuromuscular junction. But we still have step 2 out of our 4 step. The second step out of the 4 is we have to create really a full-blown action potential. All we've done so far is send a little sodium into the muscle cell, which is a little bit, which is good. It's the start of creating an action potential, but we still have to full-on create the action potential. That's the second step. And it occurs in 3 steps. The first step really is what we've already done at the neuromuscular junction. We created an end plate potential. That's creating that little bit of difference in charge. Then we have to have full depolarization and then repolarization. That's part of the action potential. So this should look familiar from action potentials. So our end plate potential, this is just kind of that small little electrical change that we just did at the neuromuscular junction. That acetylcholine that bound to those receptors, they opened them up. Some of the sodium went in. More sodium went in than potassium went out. And so it became more positive or less negative, however you want to say it. And you just created that slight change in electricity. That's what we call the end plate potential. And again, it's the end of one step, which really is the beginning of our step 2. Now, it's really getting to the point that having just a few sodium go in at that neuromuscular junction, that's when we're starting to get to that threshold stage of creating a full action potential. Because then we get to the next part, the full depolarization. That end plate potential, the fact that we had a slight change in electrical impulse right here at that neuromuscular junction, that electrical impulse is going to move. It's not going to stay right there. That electrical impulse, I don't know if I have another color for electrical impulse. We created a small electrical change right here. It's going to move along the muscle cell membrane. So you created a little electrical impulse here. That electrical impulse is going to move along the sarcolemma, along the plasma membrane. Because where, one bar lets a few people in, it tells other people to let a few more in, and then they all figure it out and the doors open everywhere. So the end plate potential spreads to adjacent membrane areas. It moves. It will create other sodium channels to open and other places along the plasma membrane. More sodium will go in, and when enough sodium goes in to become positive enough, you hit threshold, which means all the sodium channels are going to open up. And once you hit threshold, once you've opened it off enough sodium channels, lots of sodium goes in, it is a guaranteed you're going to create an action potential and your muscle is going to contract. I'm like, it's, once threshold release, you're going to make an action potential. It's unstoppable once you hit threshold, once you had enough sodium go in. The muscle fiber will contract. Now, this is showing that it will eventually move along the sarcolemma. We create that full action potential. that action potential will move along, opening up other sodium channels, creating that electrical impulse along the muscle membrane. Just keeps opening more sodium channels along the membrane so that everywhere is depolarizing along the muscle cell membrane. Now, at some point, we do have to reset ourselves. And our big thing is we created our action potential, we created the depolarization, but yes, everything that we create eventually has to get reset to be able to do it all over again. You do have repolarization. Your sodium channels are going to close. Your potassium channels are going to open. Mike, you're going to put that bouncer back at the door. You're going to let those potassium girls leave out the back door. And eventually, the sodium-potassium pump moves everything back to where it's supposed to be. Now, where everything is resetting itself, your muscles are said to be in a refractory period, which means you are telling your muscles to contract. Whether you think you're doing this or not, you are. Now, Mike, you're telling your muscles to contract, but although I say, okay, I want my muscle to contract, it just doesn't automatically happen. There is a delay, Mike, in having all of this occur. It's a really short delay, but there is a delay. And once you've created one action potential, once you've hit one threshold and you know what's going to happen, you can't hit another threshold. You have to reset yourself. You have to reset everything before you can tell your muscle to contract again. We're just going for one contraction, not two. Now, this is showing you create that end plate potential. You created that small electrical signal, that small electrical difference right at the neuromuscular junction. It's going to spread to other areas along the muscle membrane itself. So that action potential is moving along the membrane, opening sodium channels. But at some point, we're going to have repolarization. We're going to close those sodium channels. We're going to allow some of those potassium to leave so we get back to our negative charge inside the muscle membrane. Now, this should look familiar. This all happens. You're going to have sodium channels opening. So we create that end plate potential. When you open enough of them, you will hit threshold. They don't list threshold on here. And then you have lots and lots of sodium going in. At some point, before you have repolarization, you have to close your sodium channels and open all those potassium channels, and then potassium will leave until we get right back to our resting membrane potential. But you've created an action potential. So out of the book's 4-part things that happen in the neuromuscular junction, that stuff. We created an action potential. That's step 2. Those are the parts we're done. Now we're going to get to the step 3 and 4, what's known as excitation coupling. Means we're going to take that excitability we just created and we're going to match it with another ion. And then what's known as cross-bridge cycling. How do you actually shorten your muscles? Like, how do you shorten your muscles to lift something up, to move? So the excitation-contraction coupling. So you just created an electrical impulse along our sarcolemma. And I'm like, the electrical impulse is short, but what it will cause is it will cause calcium ions to move. We need calcium. Calcium was used here. Calcium's going to be moved other places as well. Now, as that electrical impulse is moving, I'm going to draw more stuff. Here's my T-tubule. Because the T-tubule goes into the actual muscle itself. And I'm like, my action potential is going to cause calcium ions to be released in other places, which really ultimately is what's going to cause your muscles to contract. The time between you creating an action potential and before you actually see the muscle contracting is a really short period of time, but it is called the latent period. Now, Mike, you still have to have stuff happening. It doesn't automatically happen. That action potential that you just created is going to travel along the sarcolemma, and eventually it's going to go down into the T-tubules. The T-tubules is still sarcolemma. It's still plasma membrane. So like it's still phospholipid bilayer all the way down. What it will do, I think your book has these as blue, is you have sarcoplasmic reticulum on either side of a T-tubule. And inside the sarcoplasmic reticulum, you have lots of calcium ions. I'll keep using calcium as my pink. That electrical signal that is traveling down the T-tubules is going to cause your sarcolemma, sarcoplasmic reticulum, to release calcium. That's calcium. So you created an action potential. It's traveling now inside the muscle itself, along your T-tubule, and it's going to cause your sarcoplasmic reticulum to release calcium. Calcium is important. So the pink is the action potential. You created it back there. It travels along the sarcolemma. It's going to go down your T-tubules. That's that sarcoplasmic reticulum on either side of the T-tubule. And it's those sarcoplasmic reticulum that's going to release calcium ions. Ultimate goal of this is we want to release calcium ions. So there's the pink electrical impulse traveling down the T-tubules, and it's going to cause your sarcoplasmic reticulum to release calcium ions, the little red balls on there. There's your calcium ions being released, because it's the calcium ions that once we release them, they're really the main ion that's going to cause your muscle to contract. Basic part of your muscle inside, along all your muscle fibers, you have myosin, our thick filaments. They have little tiny heads on them. For me, I'm always going to use them as my little arm. And you have actin, the little blue molecules up there. They're called the thin filament. They are thinner, but I can't reach the top of that. Set my pointer down somewhere. There it is. The myosin head, that myosin head has to attach to that little black circle that's on the actin. Can it attach as right now? No, it's being blocked. And I'm like, the myosin head cannot attach to that little black circle. That little black circle is called the active site. It's because stuff happens there. But it's being blocked. The active site is always covered by something known as tropomyosin, that big orange line that's right there. It's covered. That tropomyosin covers all of the active sites along your actin. And there's a molecule called troponin that anchors it in place so that it doesn't move. Guess what calcium's going to do? Because right now, calcium can't do anything. And the myosin head can't attach to the actin. That little head on there can't attach to the actin. That calcium is going to allow that myosin head to attach to the actin. So the calcium is going to attach to that yellow molecule called troponin. It's going to change troponin shape, and because troponin is attached to that tropomyosin, if troponin changes shape, it's going to move all the tropomyosin. Now can you see the active sites? So you nod, yes. Now all of the active sites are exposed, so the myosin head can attach onto the actin. That's the big part of the excitation coupling, is we had an action potential that released calcium. So that calcium combined to that troponin, they can change its shape. And now we've just opened up all of the active sites. We've made it so you can see the active sites. And these myosin heads can attach to the active sites, all because you released calcium. Now, what they will do, this gets to like step 4, is cross-bridge cycling. The myosin head will attach to the active site. It's called active site. Stuff happens there. And after it attaches, it does what's known as a power stroke. Just moves the actual actin itself, which, if you have enough of these, will shorten your muscle, your skeletal muscle. As I say, this was all of the step. You had an action potential, traveled along the T-tubules, caused the sarcoplasmic reticulum to release calcium. Calcium binds your troponin, moved to the tropomyosin. So your myosin heads can now attach to those active sites on actin. That was your step 3. The last step is, well, great, myosin heads attached, that's when you get to the last step. Now, before we get to the last step, there are lots of channels we've talked about. There are no channels in the last step. There are lots of channels so far that we have opened and closed along the way. our first nerve impulse opened some calcium channels way up at the neuromuscular junction. The acetylcholine crossed, attached to the acetylcholine receptor, opened it up so that your sodium went in. When we opened that sodium up, it caused other sodium channels along the membrane to open up so that you could create your action potential. When you created the action potential and it traveled down the T tubules, it caused the sarcoplasmic reticulum to also open calcium channels so that calcium is released. So lots of calcium moving and lots of sodium moving. Yes, there's potassium moving, but really it's the sodium that does all the good stuff. Now, the cross-bridge cycling is, why all that calcium was important. When you don't have a lot of calcium release, so at low intracellular calcium, this means before we released calcium, the tropomyosin, that big, long, skinny yellow thing, blocks all the active sites when you don't have calcium. The myosin heads can attach to it, and your muscle fiber is relaxed, which means it's not contracted. When you release the calcium, when you release all of that calcium, so you have high intracellular calcium, the calcium binds to troponin, the little yellow blob. It changes its shape, and it moves that tropomyosin string away from all the active sites. Now the myosin heads can now bind to the actin. Now, when your nervous stimulation stops, when you're done contracting your muscle, you're done doing whatever you want to do, all that calcium will get pumped right back into the sarcoplasmic reticulum. It's a storage house for calcium. Now, your myosin heads, when they're attached to actin, it's called a cross bridge. I don't know. They're attached. It's a bridge. I don't know. But the myosin heads will keep attaching and doing this little power stroke, moving that actin as long as there's calcium present, so that the tropomyosin doesn't block the active sites, and as long as there's ATP present. As long as you have calcium, as long as you have ATP, you can keep contracting your muscles. You can keep shortening them. So Mike, you can still have that cross bridge, myosin head attaching to actin, and you can still have that power stroke. You can have that myosin head walking along the actin. Now, to detach that myosin head, after it attaches and grabs onto the actin, it needs one main thing to detach and reset itself before it can grab on to another active site. It needs ATP to detach. So we need energy to be able to contract our muscles, but it's mostly because you need energy for it to detach from the actin. So the cocking of the head, so I'm like, you need ATP to release and then cock, and then it's in a ready form to be able to grab onto another active site and do its power stroke. So we run into the problem if you don't have ATP, can the myosin head release from the actin? Nope. This is showing, I've got too many things in my hands. Whatever. There's the calcium attached, and so because calcium is attached to troponin, all those active sites are available. That myosin head, it's already cocked, it's ready to go, it's going to grab onto that actin, onto an active site, and it's going to do its little power stroke. It's going to move the actin. It's going to continue to do that, but it needs ATP to release and then cock itself to be able to do it all over again. So I'm like, we need ATP to cock. I know I'm running out of time, but I want to get to at least the next slide. Well, this is the big version of it, is what happens when we run out of ATP. It's something called rigor mortis. It's after this happens, and like, you hear the horror stories that people work in funeral homes, that all of a sudden, hours after someone dies, they move. There's a reason for that. After you die, as your cells start to break down, calcium can get released into your cells. Calcium is there to cause muscles to contract. But when you're dead, are you making any ATP? No. And I'm like, which means muscles will contract after death, but will they relax? Nope. That's where rigor mortis comes from, that after death, everything stiffens. Muscles will, form a perfectly contracted state. And I'm like, because there's calcium, but there's no ATP for the relaxation part of it. Can I tell you the answers to these next things, though? Just so you know. So if you got it down, you can just circle. You ready? Just so you circle. Orange, skeletal. Not reading them. Heat generation. G, the last one, tendon, epimysium. The myofibrils. The Y, binding of acetylcholine. The latent period. We need calcium to bind to troponin, the pink. And then, because you don't have any ATP after you're dead. There's the end. I knew it would be close. Now, things to do before I see you on Thursday. 2 things. There is a muscle quiz based on this information and based on the intro video. So if you haven't watched the intro video, make sure you watch it. And FYI, I know you guys aren't watching the videos because it only had 4 views and 3 of those were mine. Then somehow it's not, YouTube's not recording it somehow. Because I'm sitting there, I'm like, it only has 4 views. Why does it only have 4 views? 3 of those are mine when I was pulling it up. Watch the videos. Maybe it's not. Oh, I wonder, I was going to say, I'm like, is it because you're watching it in Blackboard? Not. That's why. All right, well, yay for you guys watching the video. Thank you. Oh, because it's trying to do the thing. So you have a muscle video, or a muscle quiz to take, and a bunch of you have already done it, but if you haven't, that's sensory. Unit 4 sensory online quizzes due before Thursday. So if you already did it, awesome. You don't have to think about it. You just have a muscle quiz. If you haven't, you've got 2 quizzes due before Thursday.